Hello, everyone, and welcome to the fifth, or I believe sixth installment of the Diatom Web Academy series for 2021. The Diatom Web Academy is brought to you by the Society of Freshwater Science Diatom Taxonomic Certification Committee and diatoms.org. In this episode, we'll hear from Dr. Sarah Spaulding, who is a scientist at the University of Colorado's Institute of Arctic and Alpine Research and the U.S. Geological Survey. And she's also the founder of diatoms.org, and today she'll be presenting on the genus Navicula. A couple things really quick. If you have any comments about the webinar series, or if you would like to nominate a topic or a speaker, including yourself, please feel free to email us at diatomtcc@gmail.com. Any questions you have about today's talk, please post them in the chat, and we'll either answer them during the webinar or during the Q&A session at the end. A couple other things coming up. I take it you're all here because you love diatoms, and if you want to learn more about them, on May 28th, the Society of Freshwater Science will host a diatom taxon workshop. This workshop is free. You just need to register. It's hosted by Sarah Spaulding, Paula Fury, and Marina Potapova. And also, if you just can't get enough diatom information, I believe here shortly, it's at least been posted, the Ecology and Systematics of Diatom course at the Iowa Lakeside Lab will be hosted, I think, in a mix of virtual and possibly a in-person session, depending on, I think, the progression of the virus. But I at least go over and check out the course on the Iowa Lakeside Laboratory website. Speaking of the Iowa Lakeside Laboratory website, that is a research station where there are field classes hosted, and because of that organization, it was a place that led to my introduction to diatoms, and a person who did that for me is Sarah, and so it gives me great pleasure to introduce today's speaker, who's been a wonderful mentor and inspiration ever since her introducing me to diatoms. So Sarah, if you'd like to talk to us about Navicula, we would love to hear it.
Thank you, David. I appreciate that introduction. And can everybody hear me okay? Thumbs up.
Yes.
All I can see is David. So, okay, thanks. So, yeah, I wanted to start off by saying that these Diatom Web Academy talks are an offshoot of the development of a level 2 species certification program. And our Diatom TCC, we felt like, well, we can't really just test people. We need to make sure that people have the resources to learn. And so towards that end, we've had a number of talks. Well, actually, this whole webinar is really to be able to keep this really two-way communication going. And many of these talks have been, or no, maybe just a few, have been about introducing to the diatom genera. We've had a talk on Luticula, Planethidium, Eunotia, and I felt like, wow, we need a seminar about Navicula, but I don't really know anything about Navicula. And if I sign up to give a seminar, I might have to learn something. And so what I'm going to talk about today is not coming from any particular expertise that I have about Navicula, but that I felt like I'm just going to take the opportunity of giving a talk to go out and learn some things to share with all of you. So please feel free, if you have more insights about Navicula than I do, to chime in on the chat. And also along those lines, I invite any of you to commit to giving a seminar about a diatom genus. Go out and learn something, and so that you can share it with everyone. And that's really in the spirit of these Diatom Web Academy talks, that this is a community two-way conversation. So with that, I'll get started. Right, what do I do? Okay. All right. So really, the problem is that there's so many diatom species. Really, within the genus Navicula, something like 10,000 have been reported, both marine and freshwater, with 900 reported from North American freshwaters. So how do we even begin to talk about this genus? In this presentation, I'm just going to start with an introduction. And I hope by the end of the talk that all of you have a good sense of the real Navicula and kind of a gestalt for what this genus is. So kind of my outline for the talk is talk about this first diatom that was recognized and the type of the genus, what naviculoid means, and features of the real Navicula, and then talk about a few of the species in North America and their habits. So this is a page, this is a scan of a page of Mueller, 1786, for over here, Vibrio tripunctatus. This is the very first diatom that was described within Vibrio by Mueller in this reference. And here's an image of what this thing is. Vibrio tripunctatus, Mueller, 1786, page 52, plate 7, figures 2, A and B. This is right here, what we're talking about. And it just happens on the very next page was, anybody can remember, recognize this genus, Basilaria. Came next, Vibrio tripunctatus was first in the list, so it was the first diatom described. And here it is, figures 2, A and B, as the first diatom that was described. And so it later was put within the genus Navicula by Bory in 1822. And this Navicula tripunctata serves as the genera-type, or the type of the genus Navicula. And so what is this thing? If we wanted to think about it in terms of the hierarchical classification, I listed out just one. There are a number of different classifications. And the genus Navicula is within the family Naviculaceae, the order Naviculales. But a lot of times we come into seeing this term naviculoid, and that simply means navicula-like, that it's sort of like Navicula, that it has a Navicula sort of a shape. And all of these genera here, Criticula, Staurophora, and Vecadea, Haslea, all of these genera have been included in the genus Navicula at some time. And so there was, originally, many of these were described as their own genus. Houston combined them into Navicula, and then there's been a subsequent splitting things apart again. So these hierarchical classifications have sort of changed with the fashion of the day, and now they are really being separated and recognized as distinct enough that they ought to be within their own genus. So this real Navicula, the real Navicula, is what has been called within this section, Naviculae lineolate, describing the striae as being finely and transversely lineate. So a defining character, in fact, really the defining character of Navicula, so Navicula in the strict sense, is lineate or really, which is the same thing as lineole. So that's these things here. These are lineole. These are really that are elongate in the apical direction. And you could also call them lineate or really equals. They're same thing. And this is when you see that statement, Navicula sensu stricto. Navicula in the strict sense means these guys. And so that really is the diagnostic feature. And then, but I'll go ahead and say some of the other characters of the genus Navicula. So typically valves are lanceolate, linear or linear elliptic. So this kind of shape. Of course, they're symmetric, bilaterally symmetric, so symmetric to the apical axis, and also to the transapical axis. And the pattern of the striae is variable from parallel to divergent. If we look close here, these are a little bit, they radiate from the center, so that also means divergent. Too often, look at how these change direction. They're radial here, and then they're kind of almost parallel to one another here, and then they become convergent at the apices. That's kind of a Navicula thing to do. And I'm just trying to see the top of my screen. So it says something at the top, but I can't read it. So externally, the proximal raphe ends are straight. So that's right here, proximal raphe ends. These are, so the raphe ends near the center, they're either straight, a little bit expanded like these, or recurved, so they have some sort of hook. The terminal raphe fissures are strongly curved, and the striae extend around the apices. And this is not something that you can see in most species. Some of the larger ones you can, but it's really a feature that's visible under the electron microscope. So the raphe itself is straight. It goes all the way directly through the valve. It doesn't have any undulations in it, and simple. And oh, that's what I missed in the previous. That it's straight and simple, and that's shown here. Same features I went over. And then in some species, the central sternum, so this is the sternum, is raised externally. You can see this kind of more of a thickening to the sternum. And then internally, this sternum usually bears what's called a longitudinal rib on the primary side of the valve. And what this looks like to me is just some kind of asymmetry on the internal view. So here are the internal proximal raphe ends, and there's just some, asymmetric, something asymmetric. And this is what Cox calls this longitudinal rib that's on one side of the sternum that extends through the valve. And that it's variable about how noticeable it is. It's something that you can see in the light microscope if you are focusing up and down and looking for it. And that's another one of the giveaways that you're in the real Navicula, of seeing this kind of slight asymmetry to the structure. And just to review a little bit, so we can go back and understand the position of that longitudinal rib. So when the silica of a diatom is laid down initially with during mitosis, first the central sternum is formed, and then from the central nodule, and it comes around and the silica is laid down first on the primary side and then on the secondary side. So this is how we define the primary side, as being the first side that was started in the ontogeny of the cell. And then the silica closes. So this becomes the primary side of the valve, and this becomes the secondary side. And now, how can you even tell the difference? Well, you can, because in many diatoms, you'll see something called this little blip, this little place where the silica came together. It forms something that we call the Voigt discontinuity, which is the same thing. Diatomus, we have to have like at least 3 words for every term, and sometimes more. We've got 3 terms for this little mistake in the formation of the valve. The Voigt discontinuity, the Voigt fault, or the fault irregulaire all mean the same thing. And that, okay, I'm just going to back up a little bit because we can see those things. Look over here. We've got these striae all kind of lined up nicely. And then over here, there's a little blurb here, a little mistake. And that's a giveaway that this is the secondary side. This is the primary side. And so we can see that this rib, this longitudinal rib, is on the primary side of valve. Okay. Now, another Navicula feature is that internally, you got to look close here, the areli are occluded or blocked by fine plates, which are called hymens. And then the raphe terminates in a helictoglossa. That's not, lots of genera do that, have this structure here, which is the end of the raphe system. Okay, then in live cells, the real Navicula, here's one right here, has 2 plate-like chloroplasts per cell, and that 1 on each side of the girdle. And this is a valve view, so we can't really see what's going on with the pyrenoid. And all these other things are not Navicula, right? They're, they have very different chloroplast structures. Here are a bunch of Navicula live cells. Now, these are not Navicula tripunctata. I didn't have a really good picture of tripunctata, but this kind of illustrates some of the points of what Mueller was saying if we just pretend a little bit. These 3 points with that tripunctata 1, 2, not that black thing, but this round thing, 3. Does anybody know what those 3 points of tripunctata are? You want to put it in the chat? I can't see the chat, but go ahead and write what you think the 3 points of Navicula tripunctata are. Maybe Sylvia can tell me if anybody puts those out there.
No, liquid drops is one answer.
Yes, lipids. Lipids are these things, these fat storage products, and then nucleus right here. And it kind of depends what level of focus, whether you can see that. Here's a nice level of focus on the nucleus. And the lipids are storage products of diatoms. So in this next part of the talk, I wanted to just go over some of the habits of Navicula, where they live and what they do. And so I just pulled together some from a recent USGS survey of rivers. I found out that there were 130 species of Navicula out of these surveys. And one of the things that Navicula does, just like every other, we look at all the genera together, is they show this really characteristic pattern of occurrence with a few dominant, very few dominant species, and this long tail of lots of rare species. So in the recent surveys, Navicula cryptotonella was the most common, with Navicula tripunctata second in line, Navicula germanii, and down on the going in the order of occurrence by percent. So this is 10% of the Navicula in recent surveys were Navicula tripunctata. So really one of the more common species. And then I put colors on these, which I was a little bit nervous to do, but felt like, okay, we just got to get in here and see what these, what the characteristics of these species are. And these are coded with a biological condition gradient category based on, and these are posted on diatoms.org, and they came from, were they originally derived from the state of California's biological condition gradient project, and with the BC1, okay, derived from California and then expanded upon based on USGS and EPA surveys. So in this schema, looking at species and what their characters are, whether they're BC1s, really the specialist species that may be with very narrowly endemic to a particular region, BC2 being highly sensitive species, 3s being sensitive and widespread, 4s as just not discriminating. And oops, this last one, red, this is supposed to be a 5. That's a mistake. 5 as really being tolerant of worst kinds of conditions. So I color coded these along that line, along those lines, and just found most of these are really the indiscriminate taxa, that they can kind of tolerate a lot. They're not particularly valuable in determining what the conditions are. Well, no, they are. It's like more like not great. And I went back, thinking about this, went back to Potapova and Charles in a nice little paper that was in one of the proceedings in 2002. Common diatoms are usually considered superior to rare or narrowly distributed species as indicators of environmental condition, because there's ecology is better known or easier to quantify. So these are the ones that we can actually construct these optima and tolerance, because there's a lot of them. Rare diatoms, on the other hand, are often confined to oligotrophic or less polluted waters. And they're possibly really valuable because their sensitivity, but we don't have enough of them to be able to characterize them. So we're in this kind of loop of not being able to characterize the species that are really valuable. And so I wanted to kind of look at Navicula in that sense. Is that something we can say about Navicula? So what about the rare Navicula? I took that tail of the plot that I showed you over here. What are these last ones out here in the last 120 to 130 least common taxa? And these who they are, a bunch of them we're not able to put names of them on, and these were very rare. They weren't characterized on by California or within diatoms.org, except for Navicula pasta. So I'll come back to that in a little bit. And so then I thought, why don't we just get to know some of those species and compare them between the ones that we know well and the ones that we don't know well. So here's one of the ones that was actually described by Potapova and Charles, Navicula geronamensis. And this was one of the rare, possibly endemic diatoms. And oh my gosh, it really looks a lot like Navicula tripunctata. How does it differ? And it's got, just check out the central area here. It has a really strong asymmetry to it. You can see that in the electron microscope as well, that there's a strong asymmetry to the central area. Again, this one is characterized within this EC2 grouping because it's a narrowly endemic, compared to Navicula tripunctata, which has more of a symmetric central area. So part of this might be too becoming, how do we become aware of these more these species that are not so common? And oh, look over here. That's the Voigt fault. Look at that mistake right there. So that's the secondary side of the valve. That's the primary side of the valve. Couldn't resist. Here they are side by side, Navicula tripunctata and Navicula geronamensis, that they have some differences in size, but size by side, you can see more of that difference in the central area that becomes more distinct. Navicula gregaria, another really common diatom, again within the BC4, not particularly discriminating, found in, when I see this diatom, I feel like, oh, that doesn't look like such a good site to me. But I wanted to compare that to a comment that Martin Kelly made in his blog, Microscopes and Monsters. In Britain, Navicula gregaria is one of the very most common diatoms, and it shows up from really clean rivers to really polluted streams. And Martin suspect that these groups, Navicula gregaria, Navicula tripunctata, and lanceolata, are really more influenced by physical than chemical conditions, with really all they need is some cool water and well-lit conditions. So this is more of, we've got a lot to learn. One of the species that shows up in the California and other studies as being rare and valuable species is Navicula rhynchocephala, which I think is really a beautiful diatom. I love it when I see this, this taxon. And one in the of the smaller one, Navicula gersii. Can't recall if Lauren Balls described this, if or not. It is found in the Pacific Northwest. And then finally, Navicula hasta, which was one that showed up in the really good water quality conditions. And this is from a page that's in progress. It's really almost ready to be opened on diatoms.org. So here's a little review. Can you find the real Navicula in this image? Or none of the above? Is it, if this were A, B, and C, which one or none of them is the real Navicula? If anybody wants to go ahead and put that in the chat. Let's see what everybody says.
Exclamation point.
Exclamation point. C, exclamation point. The real Navicula. You have the gestalt for the real Navicula of picking that out between the Nidopsis and Pinularia. Great. Thanks, everybody. And finally, I want to just come back to the point, because it's emphasized again by my little excursion into the genus Navicula, that we failed to discover the biodiversity of species in this country because we have this, I think, really strange allegiance to fixed counts as a means of analysis. And this plot is from Meredith Tyree's thesis, looking at how much doing a 600 valve count uncovers the diversity of a sample. So here she's counted 600 valves in a low diversity site, medium diversity site, and high diversity site. And in the low diversity site, you get better than 95% enumeration efficiency. But in a high diversity site, there are a whole lot of taxa that are being missed. And I think that this problem just continues to, that we come back to this with Navicula, with being able to even characterize what species are here and what their environmental tolerances are. So this is just, my coming back to the point of, I think 600 valve counts are dumb. I think we need to find another way to do analyses if we're really interested in biodiversity and really characterizing the condition of rivers and lakes in this country. So I will end with a plug for the workshop that we're holding on May 28th. And we will be making species pages for diatoms.org. And everyone is welcome to join. We're going to take some images that are already ready to go, or you can bring your own, and you're going to learn how to load images on diatoms.org and take species pages to the point that they're ready for peer review. So please contact me or Marina or Paula for more information, and you can stay posted about the workshop that's coming up in May. And thank you for joining today. And I'll end with, can you tell me which side of this diatom is the primary side and which is the secondary side?
Thank you very much, Sarah. We have an answer. Top is primary.
And Liz got it. Right on. And if anyone has any questions, feel free to type them in. And I think I have some more quizzes, and you can, test your knowledge of Navicula, sensu, stricto in the chat. Or you can put a comment or question about anything here.
Rob says, yes, this is a Navicula.
Nice going, Rob. Okay, I can see Tom asked, how far back in the geologic record is Navicula? Thank you. I'm going to have to get back to you on that. It was something I meant to put into this talk and I didn't get there. The place that I was going to look was Mary Ellen Benson did a nice doctoral dissertation on Eocene diatoms actually in Colorado in the Florissant Formation. And in her work, she looked back at the first occurrence of really every genus that she could find. Off the top of my head, I don't remember seeing Navicula in her Eocene materials, but she would have mapped it. So I will look that up and respond back to you, Tom, on that question. It's a really good question. Thank you.
Here's a question from Kyle, a general question about size ranges listed on diatoms.org. Where do the size ranges come from, and is it only North American data sets?
Thanks, Kyle. Okay, wait, did everybody ask what? Okay, put in, is this Navicula? Because then I'll put on another one. Okay, so the size ranges, okay, thanks, Josh. This is real Navicula. How about that? Size ranges originally, so this is a little bit of a story. The Academy for a long time would measure diatoms and say whatever population they would have, measure 10 or 20 valves of the diatoms and publish biovolumes. And there's been a lot of back and forth about biovolumes of diatoms in particular, because it's kind of hard to tell the biovolume of a diatom, because a lot of times within the valve itself is vacuole, that there can be very little cytoplasm, and that it's really a variable feature, that the size of a diatom does not necessarily represent the biovolume. So, and also knowing that diatoms have a huge range in size, in diatoms.org, I started putting in the size range as being orders of magnitude. So because, sorry, I'm going to use this word, slop, because there's so much slop in the way that of measuring diatoms, that it really doesn't make sense between the amount of vacuole and this amount that the valves change of their lifespan to using those, being very specific about it. So in diatoms.org, those values are at orders of magnitude. So they're 100 cubic microns, 1,000 cubic microns, on up to 10,000 cubic microns. And so those are really quite of a broad range. Yes, they're based on North American data, although I expect that there would not be a whole lot of range, because they're set at orders of magnitude. And another caveat to that, though, is there are some taxa, like what was Somatopleura solea, we found in Iowa, incredible size range of that taxon. And so it actually spanned 2 of those orders of magnitude in diatoms.org. And we have been developing a multimetric index, and my colleagues told me that something couldn't be in 2 categories, and so they've been reduced down to 1. And so there's even more slop in what those measures are. How's that?
So Sarah, what you were talking about just now are the size metrics, right? The ecology tags.
Yeah. Oh, that wasn't even Kyle's question. Oh, size range question. Okay, sorry, wrong question. Feel free to interrupt. Yes, they are only North American, and they're only what the contributor measures. And they are subject to change, though. When people find a broader range, we expand that. So if you find that you have you think that you've got a broader range for Navicula aurora, write to us with those specimens, and we will expand that range. Thanks, Sylvia, for putting the information to contributors about what our, sort of rules and parameters for that range are.
Sure. And I know your headspace was in those size metrics, because I sent you emails.
Because you were asking me, Sylvia.
Kyle says he finds larger Navicula rhynchocephala specimens. So,
okay, well, send me, send some images, size range, and we will, I typically go back to the person who made the page for them to confirm the ID, and then we can add them to the page, some of your images, and expand the size range. And I really appreciate that kind of feedback, because that's what we need. We need all of you to give that feedback coming back, to improve the resource.
So Rob has made a stab at your question here, your quiz question. He says it has lineolate areoli, but looks like they are in alveoli. So pinnularia, question mark.
Yeah, question mark. This is a real navicula.
Ooh. Tricky.
It's in the Navicula aurora, Navicula reinhardtii group. And why else can I tell you that? If it was pinnularia, you wouldn't have the lineate or really, you know, you'd see the little pores. Okay. Okay, thanks for that question, Kyle.
Sarah, I'll ask you a question. Maybe others may have this question. So there are lots of Navicula or Diatom species that used to be Navicula. That's an older synonym. So is it wrong to call them Navicula?
Okay, so you mean, Sylvia, like if all those things that were in Navicula, wait, where are they? These guys, is it wrong to call these things Navicula?
Yes.
If you use the name, if you use the authorship of the name, so this was, it's Hippodonta capitata. It was Navicula capitata. I'm not sure, maybe it was Houston. I don't know, that whoever described it, if you use Navicula and the author of that name, then you would be correct in referring to that taxon. So it's really not a problem as long as you include a author of a name. The names that you choose to use kind of reflect your knowledge acceptance of the hierarchy, and so it's still a correct name.
Thank you. We have a question here from Jiwoo. Can I know the species name of the diatom that you had previously shown? So we know the genus of that Aurora.
It's in that, it was in that group, Navicula aurora, and Navicula reinhardtii group.
So
which, yeah. But I don't know the species name.
If you wanted to figure out the species name, what would be your process?
So my process is I need to see it in different views. I think it's pretty hard to identify a diatom just from a SEM. I need to see it in the light microscope. I need to see the size range of it. And then I sort of adjust my brain so I understand what it looks like in the scanning electron microscope. It's really a way of seeing, and I think it's something that you have to develop, of going from looking through a microscope and being able to focus on up and down and understand what a diatom looks like when you can see through it, and then what a diatom looks like when you see the surface features, which is what you see in a scanning electron micrograph.
There's a question from Gina. What's the publication containing the plate you're showing right now?
That is from Freshwater Algae of North America. The book, the big thick volume that's edited by Ware and Sheath. And this is the chapter on diatoms. There are, there are a number of plates, and there are keys to the genera in that volume.
Okay. Oh, here's a suggestion here. Can we have that presentation, the PowerPoint?
Please. Thanks, Anna. Yeah, Sylvia, if you have a place to post presentations. Okay, here's a question for everybody. Who would like to give the next, who'd like to sign up to give a genus presentation, and which one? You can do one on this list or somewhere else. I'll send you a diatom t-shirt. Matt Ashworth just volunteered. I'm not sure if he's on. Thanks, Frank. Thanks. Oh, it's, come on. It's fun. Okay, well, you can think about it. You don't have to have any special knowledge. All you have to do is be willing to learn, and it's kind of a fun exercise to dig into things. Yes, Gina, you can volunteer Rex for the genus that he's named for. Let's see, some of the ones that we have coming up. Etsy Jovanovska will be presenting on Diplonis, and Paul Hamilton will be presenting on Nidium. And so if you're not ready to commit now, that's okay. But think about it, because it's a great opportunity for you to dig in and learn about a group. And as you can see, there's lots I don't know about this group, and I did it anyway. So it's also kind of in the spirit that I hope that we get into on these webinars, is that we can learn from each other, because there's so much that we still need to learn about diatoms. So everyone is fine about volunteering somebody else. What about you? You get a t-shirt, Diatom t-shirt. So
here, Sarah, that got a little lost. Can you use ecology to decide to separate two morphologically similar specimens?
can you use ecology to separate two morphologically close specimens? The example that comes to mind is are some of the centrex within Olicosyra and within Stephanodiscus, that there are very much differences in levels of silica deposition that can indicate conditions. But I'm not really thinking about that in terms of some of the other groups. Maybe somebody else has an idea, then they can answer that question.
Well, thank you, Sarah. I think
Oh, wow. There's a, Edna would like to work on Alakasira. That would be awesome. Edna, I think you're working in lakes too in the Northeast, and there's some wonderful Alakasiras. And be glad to share images if you'd like to put it an Alakasira talk together and you have some other votes for Alakasira. So I appreciate everyone going along with this, and it's been fun to have a nice interaction with all of you.
should we end it there, Sarah?
Sure, that sounds fine. Let me know if there's something. I'm going to follow up on the appearance of Navicula in the fossil record of the information that I have. I'll write it to you, Tom, and I'll also go ahead and post it on the page with the recording for this talk. So thank you, everyone. I appreciate your joining, and it's been fun.